Hello everyone. Today we will be discussing about acid-base disorders. Acid-base disorders is one topic that confuses many students, but once you understand the logic, ABG interpretation becomes extremely simple. Let's break it down today. So moving on to the specific learning objectives of today's session. First we will be learning about the sample collection. In this we will be discussing about the type of sample which we can use for ABG analysis, anticoagulant, and also the precautions to be taken during collection and transport of samples. Then we will be classifying the acid-base disturbances into four major types. They are metabolic acidosis, metabolic alkalosis, respiratory acidosis, and respiratory alkalosis. In this particular video, we will be discussing about the interpretation of all these types of ABG disorders, and I would like to share 4 more videos regarding each one in detail. And in that videos, we will be discussing about the causes, pathophysiology, and compensatory mechanisms of metabolic acidosis, metabolic alkalosis, respiratory acidosis, and respiratory alkalosis. When we discuss about metabolic acidosis, we will be learning about anion gap and its normal reference range. Then we will be explaining the importance of anion gap in evaluation of metabolic acidosis. Moving on to the sample collection for ABG analysis. The type of sample which we are using for ABG analysis is arterial blood. This is obtained by percutaneous arterial needle puncture. This is the most commonest way, or you can get it from indwelling arterial catheter. Now, what are the preferred sites of arterial puncture? The most commonest site is radial artery. Apart from radial artery, the samples can be collected from brachial artery, femoral artery, axillary artery, and dorsalis pedis artery. In infants, the sample can also be collected from temporal artery. Now, we are going to discuss about the anticoagulant which can be used for the sample collection. So, the anticoagulant of choice for ABG samples is heparin. So, as we all know, heparin tubes are green top blood collection tube which we are using in the biochemistry laboratories. So, this can be used either as lyophilized dry heparin, which is a preferred method of collecting the sample, or we can go for heparin solution. But if heparin solution is used, the syringe should be flushed with heparin and excess solution should be removed. If residual liquid is there, then it can dilute the sample and alter PO2 and PCO2 values. Now, what are the precautions taken for sample collection and transport? Collect the sample under strict aseptic precautions. Avoid contact of sample with air. Transport the sample immediately to the analyzer without any delay. As we all know, ABG analysis is a type of POCT testing, that is point of care testing. So, if there is any delay in the transport, there will be oxygen consumption and carbon dioxide production by WBCs and platelets. Now, we are moving on to the core topic, that is acidosis and alkalosis. Acidosis, any abnormal process which tends to decrease the pH of blood. Alkalosis means any abnormal process which tends to increase the pH of blood. Now, before going to interpretation of ABG analysis, we have to learn about some reference ranges. three reference ranges you have to by heart. The first one is pH of plasma, that is 7.38 to 7.42. HCO3 levels, that normal is 22 to 26 millimole per liter. PCO2 values 35 to 45 millimeters of mercury. Along with these three reference ranges, if you know an equation known as Henderson-Hasselbalch's equation, it will be very easy to interpret ABG parameters. So what is Henderson-Hasselbalch's equation? pH equals pKa plus logarithm of conjugate base by acid. So as I said earlier, if you know the three values, that is pH of plasma, HCO3 levels, PCO2 levels, and this equation, Henderson-Hasselbalch's equation, you will be able to interpret all the ABG reports. Now few words regarding the compensatory mechanisms, or the mechanisms adopted to resist the changes in plasma pH. So the normal plasma pH is 7.38 to 7.42. So what is a first line defense mechanism? It is blood buffer. So the action will happen within seconds. Now what is the second line of defense mechanism? It is regulation by lungs. This happens within minutes to hours. Now the final or third line regulation is done by kidneys. It happens in days to weeks. So kidneys are able to excrete excess acid or alkali that are produced in our body. Now I just want to highlight two very important points, that is, first one, when there is metabolic disturbance, either it can be acidosis or it can be alkalosis, there should be respiratory compensation in our body. So once again, if you have metabolic disturbance, either metabolic acidosis or metabolic alkalosis, we are expecting the primary compensation from lungs. Likewise, when there is respiratory disturbance, either it can be acidosis or alkalosis, there should be a renal compensation. So once again, if the patient is having respiratory acidosis or respiratory alkalosis, the body has to compensate with the help of kidneys. So these are major 2 points you have to understand before interpreting and writing about the compensatory mechanisms of ABG disorders. Now we will be discussing about acid-base disorders in detail. So here, as I said, we have to know Henderson-Hasselbalch's equation, pH equals pKa plus logarithm of conjugate base by acid. So the value for conjugate base by acid, the concentration, it remains a constant and the value will be 20:1 for bicarbonate buffer. So whenever we are discussing an acid-base disorder, it will be better if you start the discussion with bicarbonate buffer. As we all know, the conjugate base is bicarbonate and the conjugate acid is carbonic acid. And the ratio of these two concentrations will be 20:1. So in our body, if we want to know the concentration of carbonic acid, we have to know the PCO2 values. So H2CO3 equals 0.033 into PCO2. So this, I am changing the equation in like it will be easy for practical purpose. pH equals 6.1 plus logarithm of HCO3 by PCO2. So that means PCO2 is directly proportional to H2CO3 concentration. Now, I want to highlight if there is any change in bicarbonate or the numerator. We are expecting a metabolic disturbance in our body. Either it can be acidosis or it can be alkalosis. And if there is any change in the denominator, that is acid or carbonic acid, here we are interpreting it as PCO2. If there is any change in the PCO2, we are expecting the patient is having a respiratory disorder. Either it can be respiratory acidosis or it can be respiratory alkalosis. Now we will discuss all these disorders in detail. pH, if there is decrease in pH, that means acidosis. If there is increase in pH, that means it is alkalosis. Now, from Henderson-Hasselbalch's equation, it's very known that pH is proportional to HCO3 by PCO2. Now we are going to discuss about acidosis. That means pH is decreased. So what change in HCO3 or the numerator will produce a decrease in pH? HCO3 has to decrease. So whenever there is disturbance, change in HCO3, we are terming it as metabolic, and pH is decreased, that is metabolic acidosis. So in short, metabolic acidosis, pH will be decreased and HCO3 levels are decreased. Now, again with the same equation, we are going to discuss the next type of acid-base disorder, pH proportional to HCO3 by PCO2. So it is acidosis, that means pH is decreased. Now, what change in PCO2 you are expecting to decrease the pH value? PCO2 has to increase because both of them are inversely proportional. So in respiratory acidosis, pH is decreased and PCO2 levels are increased. Now we are going to discuss about alkalosis. So alkalosis, what change you are expecting in pH? pH should increase. Okay, pH should increase. And first we will be discussing about metabolic alkalosis. So what change in HCO3 you are expecting to make an increased levels of pH? HCO3 also has to increase. So metabolic acidosis means pH is increased and HCO3 values are also increased. Now the last one, again with the same equation, pH proportional to HCO3 by PCO2. Respiratory alkalosis. So here alkalosis means pH is increased. Now respiratory, we are expecting a change in the carbonic acid. Carbonic acid is proportional to PCO2. So decrease in PCO2 levels can cause increase in pH. So that is about respiratory alkalosis, increase in pH and decrease in PCO2. So I hope you have understood the basic concept of all four disorders from this video.